intro into this company and their genetic soil testing. Okay?
Thank you. So I was really excited when you over there in the corner asked how you can start to measure some of this stuff to improve your farms and operations. So I'm going to try to do this super fast, but a little bit of background about Trace Genomics and what we're doing. We started in 2012 when our co-founder and CEO, Dr. Purnima Paramaswaran, started in the Bay Area, and she was one of the first people to actually teach classes at Stanford University on DNA sampling in the soil, or whole genome sequencing in the soil. With that, we've got about 10 years of experience. We started in 2015, and everything is done in our lab in Ames now. We started out here in California, but now we're all in Ames, Iowa. So, how many of you have seen this barrel before, right? We've kind of been mentioning a lot about it today, how the soil biology really plays a big role into everything. Usually we see this when we're talking about nutrients, but there's a lot of other things that all work together and can be yield limiting as well. So with that, as we've been talking a lot about endophytes and microbes in the soil, there's only about 1% of them that have already been completely sequenced. So that means we know everything about them, what they're doing. We can go and pick those out of the soil. Some examples of those are like Bacillus amyloleucofaciens, Bacillus subtilis, some of the ones we've been talking about. So that means that there's about 99% of the soil that we call dark matter or dark DNA. We don't know anything about it, but they're still present in the soil. Using whole genome sequencing or metagenomics that we're doing at Trace, we're able to look at both that 1% of known and fully sequenced microbes, as well as reaching that other 99% because we're looking at those gene functions. So when we're going through and measuring all of these microbes in the soil, I don't necessarily need to know that this microbe was Bacillus subtilis, Bacillus amyloleucofaciens. What we're more interested in knowing is what they're doing in the soil. Are they phosphorus solubilizers? Are they nitrifiers? Are they free-living nitrogen fixers? Are they phosphorus mineralizers? Things like that, so that we know what they're doing and we can come with a solution with like all of these products we've been talking about today. So the important thing to know here is that we're looking at the functional genes with our technology, and when it comes to the pathogens, we're looking at both the genus and the species level. Why that's important is there's a lot of different fusariums out there. For example, I think we've got over 40 different species of fusarium in our system. So to put that in layman's terms, why does that matter? Do I want to know if I'm looking at a dog, or do I want to know if I'm looking at a Chihuahua versus a Rottweiler? Right? That makes a big difference, and it makes a big impact on how to treat those too. So included in Trace Complete, that's our product that we offer. We have over 250 different pathogens across any crop types really. So whether you're out here dealing with lettuce, tomatoes, almonds, peaches, strawberries, or you're in the Midwest and you've got corn, beans, anything like that, we have all crop types across 250 different pathogens at both the genus and the species level. And then we've also got biological product placement like phosphorus solubilizers and mineralizers and biological nitrogen fixers. And on the nutrient management side of things, phosphorus product placement, nitrogen product placement, and micronutrient management. So I'm going to try to go through these quick since we don't have a lot of time, but at the end of the day, this is an awesome technology, but what do we do about it, right? As growers, as agronomists, if I can't make a decision based on this data, what's the point? And that's why we have our reports. But as we're talking about pathogens, we have to keep in mind the disease triangle. Just because we're measuring this DNA in the soil, if we don't have the right environment for that, then it's not we're not necessarily going to see it. So we have to keep that in mind as we're measuring these different diseases. It's great to have this technology, but you always have to keep that environment perspective in mind too. So this is an example of our pathogen report. Everything is still included and in the background in our TraceView database. I can show that later this afternoon if you're interested. But this is an example showing where those measurements are. So everything's measured in a parts per billion number based on those DNA. So I could tell you that verticillium wilt for this grape example, maybe it was 0.037 parts per billion, right? But that doesn't do you much good if that's all I came back to you and said. So that's why we put it up against our benchmarks and on this bar graph. So if it was in the red, you would see, I'm trying to see if I've got a better example. If you would see what percentage that was. So sometimes I see them come back as like 3,000 or 4,000% of that benchmark. So not only were we hot, but we were 3,000% of that. And that's when you can start to make some of those actions on that too. So these are just examples of the different crop types we have that are kind of regional to you guys here. But because we're talking about endophytes and products, everything like that, this is our Trace Phos report, where we're bringing together both the chemistry by looking at Bray, Olson, and Malick on the phosphorus side, but then also the microbes who have those gene functions again for phosphorus solubilization or mineralization. So now if I know that I've got some of these really good products we've been talking about, now I know where to best put them based on the microbial levels that are out there. Same thing with Trace N. We are measuring microbes who have the functions for urea volatilization, nitrification, and denitrification. So where those high levels of nitrogen loss based on the microbes. And then where can I put products like Twin N, like we were just talking about. And same thing, we can measure free-living nitrogen fixers as well as the Rhizobia species across legumes. So those are also included all in this same single test through whole genome sequencing. And then in addition to that, these are just the reports we have. Some of the other measurements we have too are like your fungi, bacterial to fungi ratio, oxygen availability. We've got mycorrhiza. There's a whole list of them. We have 20 different nutrient cycling indicators in addition to the pathogens and everything all included. So, yeah, if you have more questions.